Hello, biology students. This is Mr. Friedhoff. This is going to be chapter 14, and this is kind of an odd chapter. It's about biotechnology. What biotechnology is basically how we use technology in biology. But what we see is we can use a lot of things that we're learning about, such as like DNA going to RNA to protein, which is the central dogma, and we can apply those to our lives and actually kind of help us learn things about different fields like forensics or genomics. And we can actually use that technology to really further our understanding of just life in general. So what biotechnology is just basically utilizing our technology and developing better products that will help us with our daily lives. And there's a lot of different areas. The reason why I say it's kind of a weird chapter, there's a lot of different areas of biotechnology that don't really like fit together. It's not like there's a story that's told. We're kind of one section here is going to lead into the next section, which leads into the next section. Basically, what happens in this chapter is there's a lot of just cool parts of technology, and we're going to go over each part just kind of in a different section. But they somewhat might relate, but they might not. It's just a really cool chapter to see all the technology out there. So with that being said, we're going to get into 14.1. All right, 14.1 is going to go over something called gel electrophoresis, and that's a big word. First, before we get into gel electrophoresis, let's talk about PCR. PCR is something called polymerase chain reaction, and this is just basically a machine that's going to make many copies of DNA from a small amount. So basically, this acts like a replicator of DNA. Now, I know you're probably thinking, like, who cares? How, DNA is DNA. We can make DNA ourselves. If we have a crime scene, we're talking really, specifically about forensics, and in that crime scene, we only have a little bit of DNA, but we want to test it a bunch. We don't want to ruin that DNA. So what we're going to do is we're going to take that sample of DNA and we're going to copy it a bunch using this PCR machine. So again, basically what this does is it just makes many, many, many copies of a small amount of DNA to enable us to test that DNA in many different ways. What we then do is we run that DNA through something called a gel electrophoresis machine. And this machine is going to separate the DNA based on size. So what this is used by is they're used by forensic scientists who are trying to maybe like catch a criminal or catch a murderer or catch somebody that's a suspect. And they use these machines to basically, again, just separate DNA. So remember that each person is going to have a unique set of DNA, and we call these kind of repeats in the DNA mini satellites. And these are just fragments of DNA. They're going to be separated based on, you can see here, based on the gel electrophoresis. The ladder on the left-hand side is the already pre-known fragment lengths. You can see here, these are the fragment lengths. They're already pre-known. And what we can do is we can separate the DNA of like 4 different people. Like you can see Jenny, Bob, Mike, and Lisa. These are the like suspects maybe in a crime. And they're going to separate the DNA based on length. That's what the machine, that gel electrophoresis, does. What happens is, again, we're going to take this DNA that we have, and we're going to cut it out of our like the cells, and we again use these restriction enzymes to basically just cut the DNA. And we place that DNA into a gel plate. You can see those gel plates. There's little wells in there. And what those wells will do after a while, after an electrical current sent through them, is they'll separate the DNA based on length. So what happens after the electrical current is placed through the gel electrophoresis is, again, it's going to separate that DNA based on length. What happens is the largest bands don't travel very far, while the shortest bands travel the furthest. So you can see here, the smallest bands are going to be the bands that, again, travel the furthest, while the largest ones don't travel very far. So if we look here, again, here's our ladder that we already know. This is the pre-known DNA. Each person is going to have a unique set of these bands, or these unique set of DNA lines. So these DNA lines will, again, identify a person based on their unique DNA. So this is what it looks like. You can see the wells here. These are the wells that separate the DNA. And you can see they are being pulled by that electrical current by their size. You can see the shortest ones go the furthest, and the largest ones don't go very far. Here's another way of looking at it. You can see, again, the longer fragments travel not as far as the shorter fragment. This is what is actually happening in that gel plate, is the small fragments can travel further than the larger ones. So again, smaller fragments travel the furthest, larger fragments don't travel very far. This is what it looks like. This is what those bands are representing. They're just representing different fragments of DNA. Now, what this allows us to do, before we get there, let's look at this little quiz here. Which person has the longest fragment? So person one, two, or three? Well, person one has the largest fragment right here. That's 1,200 bases. Which person has the shortest fragment? That'd be person three. You can see their shortest fragments right there. How many fragments do let me go back here. How many fragments are the same length between person one and person two? So if we look here, person one and person two, this fragment's the same and this fragment's the same. So two fragments. Our last question, how large is the fragment that all three people share? So this fragment here, all three people share, and that fragment is 1,000 base pairs. All right, in our next section, we're going to talk about something called genetic fingerprinting. This is exactly what we just talked about in the last section with our gel electrophoresis. However, now we're going to use this in forensics. Basically, what we're going to do is we're going to look at how those unique bands or how those unique DNA kind of like lines allow us to identify a person based on like whether or not they share those bands. So again, you can see here we found DNA at a crime scene and we have 3 suspects. What we can do is we can match those up. You can see here, like suspect 2 is the person that was at the crime. Now, this doesn't mean for sure they did the crime, but if they were at the crime scene, again, this is going to lead the prosecutors or the courts to say, hey, they're most likely the person that was at the crime scene during this crime. Let's do another one here. You can see here, bloodstain. Who is the suspect that was at the crime? So again, we're going to match up the lines here. You can see suspect 1 isn't it, suspect 2 isn't it. But if you look at suspect 3, they have all the same lines. I know they're different colors, but this helps us see what we're looking at a little bit differently. So again, we have the bloodstain here. We have the victim here. Who, which are the 3 suspects, who is the person that was at the crime scene? So what we have to do is we not only have to look at the victim, because the bloodstain might be the victims, but you can see here, well, it's not going to be the victims, it's going to be suspect 2. That's a video we're not going to watch. So what we're also going to do, let me go back. What we're also going to see is that genetic fingerprint can also be used with parents because again, you share half of your DNA with mom and half of your DNA with dad. So again, you're going to have half the DNA fragments that your mom has, and the other half are going to come from your dad. You can see here, this child has half of the fragments that come from mom and half the fragments that come from dad. Like in this case, you can see the mom here has all of these fragments. The baby shares some of the fragments, but like this fragment doesn't come from mom. So that means it has to come from dad. So that means dad 1 can't be the real dad. So we look down here. This one comes from mom. This one comes from dad. Again, we already rolled out dad 1. Now this one rules out dad 2. This means dad 3 is this child's real father. If we do another one, we can again see the mother's fragments. We can see the father's fragments here. This means that the person father 1 is the true father here, not father 2 or father 3. All right, hello everyone. We're going to talk about section 3 in chapter 14, which is about genetic modification. This is where we're going to use technology to either insert DNA or edit the genome of an organism. So we're editing DNA basically. So again, in this chapter, we're talking a lot about just different technologies and how they're being applied today to modern problems. And one of the ways that we've done this is using something called CRISPR-Cas9. Now, years ago, we thought this was going to be possible, but in 2012, I know it doesn't seem like that recent, but in the sense of like DNA in the past 100 years, this is really recent. We identified this set of proteins in bacteria cells that can edit the genome of bacteria. And what we're using now is that same system in our cells. Now, what we can basically do is edit genes and edit the DNA of somebody. And this is a big technology that we can use, but we're also kind of a little bit scared because, again, when we start looking at genomics, we have to understand, okay, what can we do? What can we not do? What shouldn't we do? So there's a lot of ethical things that we have to kind of tiptoe around to make sure we're not causing problems where there aren't problems already. So each of these we'll watch a video in class, but just since we're just doing the lecture here, we won't get to those videos. Another type of genetic modification is called gene therapy, and this is where a virus is going to be used as a transporter for a healthy gene. So again, let's say somebody doesn't have a healthy gene, they have like cystic fibrosis. What they can do is they can use a virus to actually insert a healthy gene into a cell. Now, I know you might be thinking, well, won't that make the person sick. Well, these viruses have been edited so they don't actually make the person sick. And what will happen is, again, they'll get the healthy gene so they can produce a protein that they need. We've been using this for years and years and years, and this is a really cool way to help somebody that may have a deadly disease. Another type of genetic modification we're going to talk about is something called a GMO. And GMOs are genetically modified organisms. This is where we take the DNA of a plant per se, and we edit it to make it either healthier, we make it produce more food, we just make the plant better overall with more beneficial traits. And when we do this, we can really save lives. For example, golden rice has been inserted with a gene to make vitamin A, and this combats a vitamin A deficiency in these Asian populations. You can see this golden rice again has just higher amounts of vitamin A. BT corn produces much more sustainable corn because again, it's going to kill the pests that eat the corn, and it's non life threatening for us, or it doesn't really affect us. So again, this BT corn produces just more healthy and a higher yield product than just regular corn. Like showing this image of what back in the 50s what we had to do to produce insulin for people that are diabetic, and now we use bacterial insulin because it's a lot more ethical and it's a lot just more efficient and better. So there are some benefits and some drawbacks from GMOs. Benefits are obviously better yields of crops, more nutritional values of crops, or crops that are resistant to droughts, floods, and frost and diseases. But there are also some drawbacks where, like GM companies, these genetically modified companies that own these patents on the genes, they really make it harder on smaller farms and smaller just like farm just in general, and these GM crops also may harm non-threatening insects. And this can again lead to biodiversity drops, which is not good for environments. They can also just lead to problems just in the ecosystems here, as you can see. So we have to understand the benefits and the drawbacks of using these GMOs. Section 14.4 is going to be very short. It's just about cloning. And this is where we're going to replace the nucleus of an egg with another cell's nucleus. And what happens is you create an organism that's exactly like the organism from the nucleus. So again, we have this frog here. We have an egg. We take out the nucleus. We take the nucleus from another organism and we turn it into that egg. And essentially that egg will become a clone of the second frog here. It's an exact copy. And this might be a weird thing to think about, but that's what essentially, not perfectly, but that's what essentially identical twins are. So they're just clones of each other. So this isn't something like really, really new. We did this back in the 90s. The first vertebrate to be cloned was something called Dolly the sheep, and it died, I think, after a couple years. But we've gotten much better at cloning. And I'm going to show you a video in class where there's actually companies where you can clone your dog, and they cost about $100,000 to complete that process. I know it sounds kind of crazy for us, but some people pay $100,000 to have their dog cloned. All right, lastly, we're going to talk about in our chapter 15.5 is vaccines. Vaccines are just biological preparations that we use to help our immune system. We've been using vaccines for hundreds of years, and they are safe and effective. There's a lot of misinformation out there about vaccines because of the COVID pandemic, but to assure you, there is, again, just a lot of misinformation. We're going to go over some of that misinformation here. Basically, what they do is they help your body produce antibodies. And these antibodies will help fight off viruses and other bacteria cells that try to infect us. But vaccines stimulate your body to produce these antibodies and to help our immune system fight those different diseases. What vaccines have helped us do is decrease the mortality rate of certain vaccines. So you can see here, these are certain diseases that cause in the 20th century thousands upon thousands, if not millions, of deaths. You can see after after the inception of their vaccines, what they've done. They've dramatically decreased the amount of deaths per these different diseases. Now, I'm going to go back here. Obviously, some of these diseases have started popping back up because vaccine rates have started to decrease because of this misinformation. The COVID-19 vaccine is a little bit different, but it's still effective, and it's really cool how they did this. Basically, what they did was they made a mRNA strand to make these little they're called antigens from the virus, and your body will recognize these antigens and help make antibodies against these antigens. So this is a more effective and just a quicker way to produce a vaccine, but it's still, again, very effective at fighting the COVID-19 virus.